Hello, good afternoon, everyone. I first want to say thank you to the Tohoku Forum members for the nice invitation for the event today and the yesterday, and also for the members and friends from the CFI. It's okay. From the CFI. I'm coming from Argentina, very far away, and I belong to the institute. It is called CERELA. Actually, Argentina has a government, a national government organization called National Council of Scientific and Technological Research that is constituted by maybe 800 institutes dedicated only by research, and I belong to one of them that is dedicated only to the study of lactic acid bacteria. And our laboratory has been working with the use of these beneficial microbes to improve resistance to some infections. And I will tell you a little bit about what have we done with the resistance against viral infections. As you may know, viral infections are very important, especially in high-risk population. That it means little children, elderly, and those with some compromise with the immune system. And actually, these two types of infections, diarrhea and respiratory infections, are the major killers for young children and also for young animals. In fact, maybe 30% of the little children in the world die because of these two infections. And these infections are mainly caused by virus. That is a very big problem in the world because the resistance against antimicrobial substance of the viruses have been increased. Several speakers have mentioned that there is a global concern, the increase in the resistance of antimicrobial resistance in bacteria, but also viruses are showing the same pattern. And actually, bacteria can increase the resistance to antimicrobial substance because of the high pressure of the misuse or the excessive use of a substance, but virus don't need actually that kind of pressure because they themselves are always changing and trying to survive the host. So anyway, the viruses are actually a big problem because of the higher resistance. So we are trying to find different alternatives to fight infection, especially in very susceptible hosts. And one of the interesting tools that we have is to use beneficial microbes. You know, I love this cover that was published in Trends in Immunology because it clearly shows the great influence that microorganisms have in the immune system. You have here a bacteria and all immune cells, and bacteria are actively involved in the development, maturation, and homeostasis of the immune system. So scientists have learned how to use these beneficial microbes to improve our defenses against microorganisms. For example, several research in the world have demonstrated that we can use beneficial microbes to improve the local antiviral immunity. I mention local because mainly the studies have mainly evaluated the effect at the intestinal level. But several laboratories, not so several, but some laboratories have also demonstrated that the bacteria in the gut can influence not only the local antiviral immunity, but also immunity in distal mucosal sites like the respiratory tract. So these beneficial microbes become a very interesting alternative to improve the local and the distal antiviral immunity at the same time. Among these microorganisms, lactic acid bacteria, there are a group of these microorganisms that can improve the health of the host that are called probiotics. Actually, that is the definition of probiotics. And among them, there are a lot of strains that work improving health by communicating with the cells of the immune system. And we have advanced a lot in the knowledge in the interaction of these microorganisms with the immune system. So now the people or the researchers that work in the field, we want to separate them from the other microorganisms because I think they deserve a special name, and we call them immunobiotics to distinguish from the other mechanisms of action for improving health. So in my talk, I will use this term, immunobiotics, to describe beneficial bacteria that are able to confer some health benefit through the interaction with the immune system. Our laboratory have worked for many years searching that bacterial strain that can improve resistance to infection, intestinal and respiratory infection, and we mainly work with bacterial infection like Salmonella, E. coli, or Streptococcus pneumoniae infection. And among the bacteria that we use, we found that have nice activities, we found that this strain that is Lactobacillus rhamnosus CRL, because it belongs to the CERELA culture collection, and 1505, was able to improve in vitro and in vivo models to improve resistance against intestinal and respiratory infection. So finally, CERELA, in collaboration with the government, national government and provincial government, decided to test this strain in a human trial, and we decided to give that to little children. Children attending to a community places where they receive lunch once a day, and they receive this strain incorporated in a yogurt, and then we follow them for 6 months, and finally we were able to demonstrate that the children that consume this probiotic yogurt were able to reduce the severity and the incidence of respiratory and intestinal infection. Finally, the government got excited and decided to incorporate this product in the schools, and actually this is given for free to all the children in attending the school, primary elementary school. But what is interesting from this clinical trial is that we have performed mainly studies in the laboratory by using bacterial pathogens. But when we analyze the pathogens that actually infected children during this clinical trial, we found that most of them were infected with rotavirus, influenza virus, and respiratory syncytial virus. So we had an indirect evidence that this strain was very good to stimulate antiviral immunity. So we decided to study the mechanism by which this strain stimulated antiviral immunity. As you can see here, this strain has the unique characteristic that stimulate both the local and the distal antiviral immune response. That this is not a common characteristic. We have tried and studied several immunomodulatory strains, and some of them have this characteristic and others has not. So we focus first in the local antiviral immunity by evaluating the effect of this immunobiotic strain against rotavirus infection, and we have found that actually this strain is able to interact with intestinal epithelial cells and also intestinal immune cells like dendritic cells, and improve the antiviral state by increasing type 1 interferon and interferon gamma, and also modulated in a different way the inflammatory response. For this study, we have used porcine intestinal epithelial cells in collaboration with members of the C5, because the porcine immune system is very similar to human immune system, so it's a very good model for us to study. And also you will see that in all my presentation I put this strain. This is another immunomodulatory strain. This is 1506, that it's also able to improve antiviral immunity, but only at the local level. When you give this by the oral route, you can improve the resistance in the against infection in the intestinal tract, but not in the respiratory tract. So the comparative studies of these two strains give us a nice opportunity to understand why some strain can activate the immunity in distant mucosal sites, and why other they don't have that capacity. We also demonstrated that in addition to reducing the replication of viral infection, the immunobiotic strains are able to protect against the inflammatory damage. You know, these are two important components during viral infection. One is related to the replication of the viral itself, that is the cytopathic effect, but also there is an important component of the immune system involved in the damage during viral infection. In fact, sometimes, especially in the respiratory tract, inflammation should be controlled more than the replication of the virus. I mean, if you have to choose what to control, or the replication of the viral or the inflammatory response, I will choose inflammatory response because you will have a better chance to survive. And some studies, not in my laboratory, but other studies demonstrated that one of the mechanisms that rotavirus used to induce a very severe inflammatory response in the gut is through the activation of toll-like receptor 3. Some comparative studies using the genome, the purified double stranded RNA from rotavirus or poly-IC, induce the increase of a cytokine that recruit intraepithelial lymphocyte like this phenotype, and the activation of them that inducing severe damage in the gastrointestinal tract. By using this model, we show in mice that the administration of these two immunobiotic strains actually reduce cytokines like interleukin 15, and in that way reduce the damage mediated by this intraepithelial lymphocyte. As I told you before, we also investigated how these two immunobiotic strains influence the resistance against respiratory virus. I will not show everything, but in this slide, it's like a summary of all the studies that we have done maybe for 5 or 6 years, including other strains, because other laboratories have also demonstrated the ability of some immunobiotics to improve resistance against respiratory infection. And you can see some of them are able to improve the innate antiviral immune response, created an antiviral state, improve the production of specific antibodies. And what was demonstrated lately is that some of them are able to differentially modulate the inflammatory response. Studies by evaluating the quantity and the kinetic of inflammatory cells, inflammatory chemokines, inflammatory cytokines, and the expression of adhesion molecules show that some immunobiotic strains can change these factors involved in the inflammatory response, and then beneficially regulate the lung tissue damage and improve the survival of infected mice. This study also, we have also showed that immunobiotic can influence other systems that are involved in the defense against viruses, because the immune system is not the only physiological system that is in charge to defense against virus. Another system is the coagulation system that actually it helps in the damage produced by some respiratory virus. Some respiratory virus like influenza virus or respiratory syncytial virus can interact with endothelial cells and alveolar macrophages, inducing the expression of this factor, that is tissue factor. Actually, the increase of tissue factor and the reduction of the thrombomodulin, that are the two factors that regulate the activation of coagulation, are differentially regulated to the viral infection, leading to the strong activation of coagulation and a severe alteration of the alveolar capillary barrier. This causes that the blood can go to the alveolar space. So it's very typical in the lungs of infected mice. You can see that the very light pink lung becomes very red lung because of this mechanism. We have tried, we have performed experiments to evaluate whether the immunobiotic strain can modulate this activation, and we found that actually they can reduce the coagulation of activation and the alteration of the alveolar capillary barrier, mainly through the modulation of the immune system. So, so far we are trying to find to explain how these microorganisms that impact in the intestinal tract can influence the respiratory tract. We have demonstrated that it could be related to the mobilization of some cells, lymphocytes from the gut to the respiratory tract. Here it is shown only CD4 cells, but we have also shown for the B cells, IgA positive cells, that after being stimulated in the gut, they move to the respiratory tract and they produce cytokines like interferon gamma that stimulate local antigen-presenting cells that in the face with the pathogen, respiratory or viral pathogen, they can generate a different immune response. Maybe this is not the only mechanism. Other laboratories in the world have proposed different mechanisms. For example, some of them have demonstrated that cellular fragments from immunobiotics can be released into the blood and reach, for example, the bone marrow and stimulate a steady state activation of neutrophils that in the face of infection that are rapidly and efficiently recruited into the lung. And recently, other studies have demonstrated that the immunobiotic can impact in the metabolic activity of the microbiota in the gut, and then induce the production of some substances like a small short-chain fatty acid that are released into the blood and then interact with some immune cells in the respiratory tract, changing the immune function there. So there are three options. Maybe they are acting together. We have not studied the other one, so maybe they are not. I mean, we cannot explain the effect of one immunobiotic strain by only one mechanism. We should study. So we really want to know what happened here when immunobiotic strain impact for stimulating antiviral immunity locally and in the distal tract. So we wanted to use the new generation of tools that by using genomic studies, and for that we have initiated a collaboration between several laboratories of the C5 and also the Argentina and including Chile, to study the mechanism of action of several immunomodulatory strains with different activities. And again, we use the porcine intestinal epithelial cells as the model, and by using the microarray technology, we evaluated how this immunobiotic strain can modulate the immunotranscriptomic response. We have found different in between the groups, but when we concentrated our attention in only the immune-related genes, we found that actually pi cells can generate a very complex immune response. Perhaps it was surprising because some years ago epithelial cells were thought to be only a physical barrier, but now we know that they are very important in the generation of immune responses. As you can see here, after the stimulation with rotavirus or poly-C, they can generate type 1 interferon and several factors that increase the antiviral state, and chemokines, cytokines, adhesion molecules, prostaglandin, complement factor that are involved in the defense against the viruses. But the most interesting thing for us is that actually immunobiotic strain can modulate several of these genes. Some of them are increased, for example, this antiviral factor that would explain why the children or animals that are treated with this immunobiotic strain have a reduced replication of the virus, and other changes like this one in adhesion molecules or chemokine could explain the reduction of the inflammatory damage in children and animals. And what is also interesting for us is that this strain that has only a local effect have some quantitative and qualitative differences when we evaluated epithelial cells when compared with this strain. So this can give some clues to understand the mechanism, the differential immunomodulatory mechanism of these two strains. But actually, we think we need to do this kind of experiment by using also immune cells, and we are working on that. And we also selected several genes here that that we can use as a biomarker to select new immunomodulatory strain. Because it is very easy to say that if you have a microorganism and have a very good stimulatory activity, you can use it for improve the health of human. But when you try to do, sometimes when you have the microorganism and you want to put it in a product or cultivate it in a bigger scale, sometimes it is a big problem. So biotechnologies have to be also involved in the development of this kind of product. So sometimes we need immunomodulatory strains, but that also have very good biotechnological application. So we are working in the Institute in a big project that is called the Micro Andes Project, that it's mainly dedicated to the isolation and characterization of beneficial microorganisms for the biotechnological application. You know, this is Argentina. It's a very, very big country. And from here, from the Atlantic Ocean to the Andes, you can have lands from this at the level of the sea until maybe 7,000 meters in the Aconcagua, and then from the north, very hot weather, to the south, and very cold. And if you combine altitude and weather, you have many, many different type of places in the same country. In the north region, we have unique plants and animals and also products that have a very strong influence from the Inca Empire. So our institute is trying to isolate and make a characterization of a typical lactic acid bacteria strain from this type of product, and by using these biomarkers that we found in Pie cells, try to find bacterial or beneficial bacteria with immunomodulatory or antiviral capabilities. This project is mainly dedicated to human health, but after several years of interacting with members of the C5, I also became interested in animal immunity, and I believe that we can use the same strategy to improve the health or immune health, or specifically the antiviral immune state of animals that are economically important. You know, in Argentina, the cow is the most important animal. Maybe it's related to all our foods, not only related to meat and meat products, but also to milk and dairy products. It's often found in Argentina's table. So I will not give you all the details, but we have performed a similar experiment by using a very nice epithelial cell developed in the laboratory of Dr. Hisashi Aso, that is the bovine intestinal epithelial cells, and by using also transcriptomic analysis, we found a set of genes or biomarkers that we can use to find or to select new immunobiotics strain with antiviral capabilities. And for that, we are another big project of the CERELA. This is the micropampa, you know. This is in the central region of Argentina. We call this the pampa. It's like this, only land with cows and for kilometers. And we are searching and isolating bacteria from different sources, mainly from milk and milk products and cows, and searching a new immunobiotic strain with antiviral capabilities. So by the end, I think we will have a library of immunomodulatory lactic acid bacteria that we can use for improve antiviral immunity, not only in humans but also in animals. That would be very helpful for us to develop new immunobiotic food or feeds that can improve the health of this host. I think. Thank you very much.